Hi everyone, thanks for joining me on the online version of my poster. And my poster today is going to talk about the intersection between some academic research to understand the consequence of a mutation and then its life through our high throughput discovery pipeline for a patient who has a DYRK1A syndrome. So DYRK1A syndrome is an autosomal dominant rare neurodevelopmental disorder, and it's caused by loss of function mutations within the DYRK1A gene and protein. And this leads to what's known as haploinsufficiency of DYRK1A. So haploinsufficiency is a genetic state that describes the fact that when there's one loss of a gene or one loss of a copy of a gene, the remaining healthy copy cannot produce enough gene product to keep cellular homeostasis. This is not true for all genes, as some genes can tolerate the loss of one allele, but a lot of genes like transcription factors and kinase often can't tolerate this loss, and obviously DYRK1A is one of them. So DYRK1A has a widespread role within the cell, as you can see. I'm not going to go through these one by one, but what I will do is highlight the ones that I think play a critical role, at least in the neurological phenotype we see in DYRK1A. So here you can see the first one I've highlighted is cell cycle progression. Now DYRK1A acts as either a promoter or a staller of cell cycle depending on the context, which makes it very difficult actually to understand its role within the disease, because in a certain context it can promote cell cycle, which basically means it promotes cell division, and in the other hand it can actually inhibit it. But what it seems to be with the context of neurons is that these cells fail to exit the cycle. So here you can see we have these various regions of the cell cycle or stages of the cell cycle. And what DYRK1A does in neurons is it exits what we call G1 and exits into a quiescent or a senescent state of G0. And then this helps for cellular differentiation. So the next one obviously then is cellular differentiation. And then DYRK1A here plays another critical role, which it actually promotes the differentiation of cells once they've exited the cell cycle. So individuals with DYRK1A syndrome seem to have a higher pool of replicating progenitor cells that fail to differentiate at rates that are needed during the development of the brain. And then this can affect things such as axonal transport and migration. So axonal transport and migration of these neurons is critical to forming the correct regions within the brain. And it's believed that what happens is these mature neurons that actually do get made, they do not migrate to the proper place and eventually will die. And this will contribute to the microcephaly that's seen in some of these patients. Another key factor that DYRK1A is involved in is splicing. And obviously splicing has a global effect on most protein coding genes. And I believe you may see another poster by Wei Zhang, who's going to talk to you about mutations within a splicing component that does cause a form of neurodevelopmental disorder as well. So if DYRK1A affects splicing, you can see that it'll have a significant amount of downstream consequences. And then these consequences can also be involved in all these other cellular mechanisms. So it's no wonder that DYRK1A syndrome doesn't primarily only affect the brain. You have quite a widespread phenotype when it comes to this syndrome. So our current patient harbors what we call a 1072-1 G2A mutation, and it's currently the only patient in the entire world with this specific mutation. So due to its location and novelty of this mutation, little was actually known about the consequence. So we know where the mutation is, but we don't actually know what the mutation does to the gene. Initially, it was thought that it might create a cryptic splicing event or a frame shift event. And this is where the academic research began. How does this mutation affect the gene? And therefore, how does that consequence affect the protein? So just before I get into that, I want to talk to you about the phenotype. And I'll go through this quite quickly because I'm sure you can just read it. But DYRK1A not only affects the brain, it can affect the eyes, and these are typically deep set, and they'll often have eye abnormalities requiring glasses. It can affect your upper body, so your gross motor skills. You generally have delayed growth, a short stature, and low birth weight. And obviously, primarily, one of the main regions that's affected in any neurodevelopmental disorder is the brain. So here we have microcephaly, which is a small brain size and volume. Almost all patients have intellectual disability as well as autism spectrum disorder. And a group of patients or a subset of patients would have seizures and severe speech delay. What's interesting is a lot of these individuals have severe GI tract issues. This can range from chronic constipation to food sensitivities, and new budding research in animal models thinks that this is because DYRK1A is required for proper cilia formation, and that potentially the lack of DYRK1A in these regions causes defective cilia. And in terms of the lower limbs, they often have fetal abnormalities and have an unsteady gait. So you can see that the DYRK1A gene affects the whole body rather than just a pure neurological phenotype. So work done in an academic setting before the case was accepted into n-Lorem was needed because obviously, as I said before, we initially thought that it might lead to a cryptic splicing event that would lead to a frame shift, and that would be a whole different ASO strategy. So further work led us actually to discover that the mutation leads to what's known as complete exon 8 skipping. And the reason this is because the position of the mutation, the -1, sits in what's known as the 3' splice site. So the 3' splice site basically defines the beginning of an exon, and there's a splice site at the end known as the 5' splice site. And these are coding regions that then are destined to make a protein. So the cell really needs to have a definitive start and end of an exon to accurately put together a gene. And by mutating the base from a G to an A, we've lost what we call the canonical splice site. And that was enough to trick or to confuse the cell into thinking that exon 8 was a non-coding region and then skip that region. So there's a lot on this slide, but I'll go panel by panel, just so that we can walk you through how we discovered this. So initially we did a RT-PCR and ran this on a gel, and we had several controls. So here is a neuronal control, and then here is a healthy subject fibroblast control, and this is our DYRK1A patient. And as you can see, in neurons as well as the healthy subject, there's only one band that represents the full length of what we were trying to amplify. Here you can see there's another band slightly lower in the DYRK1A patient sample, and this means that there's a region here that is missing from the full length. So we did an assay known as Sanger sequencing, and we actually found that exon 7 now is ligated or attached to exon 9 instead of exon 7 being attached to exon 8. We further investigated this through RNA sequencing, which I haven't shown today, and plots show this exact same mechanism is happening. From the diseased allele, we're getting complete exon 8 skipping. So how does this actually affect the DYRK1A protein? Well, here is a to-scale transcript and protein map. So up here we have the DYRK1A transcript and its exons that are numbered. And obviously exon 8 is in red. And what happens is removing that exon, we are going to remove this region from the kinase domain. And if you remember, I said that DYRK1A is a kinase, and this is really its functional domain. This is where it adds a chemical group onto other proteins that can either activate them, deactivate them, stabilize them, destabilize them. So without having an active region within your gene, basically what you will create sometimes is called a dead kinase, where the kinase doesn't work at all. But interestingly, exon 8 is in frame. So what that means is that it doesn't share any amino acids with its neighboring exons, and that when you remove it, you would keep the reading frame intact, so that you wouldn't have a frame shift. So we thought that we might be creating an internally truncated protein that's missing this region, as well as missing a nuclear localization signal. So then we wanted to check, well, is there reduced expression of DYRK1A compared to controls? And as you can see here in our DYRK1A patient compared to 2 healthy controls, the gene expression levels are very similar, although the patient has disease. Now, this again comes back to the exon being in frame. If the exon is in frame and it's removed, the cell won't trigger a mechanism known as nonsense mediated decay. So then we further wanted to investigate, well, where is the haploinsufficiency coming from? So we did a Western blot, which is a protein quantification experiment. And here you can see we have a healthy subject, which has quite a strong signal for DYRK1A. Then we have our DYRK1A untreated sample. So you can see that it's definitely lower at the protein level. We have a non-targeting control, and then we designed an ASO to actually skip exon 8 from the healthy allele to see what that would do. And as you can see, when you remove exon 8 from both alleles, you actually do not get any DYRK1A protein. So this means that actually the haploinsufficiency is coming from the protein, and that the protein missing exon 8 is most likely rapidly degraded or not translated at all. So this led us to our current therapeutic strategy. So because exon 8 is being excluded from the mature transcript, what we needed to do was design fully modified ASOs on a PS backbone or a phosphorothioate backbone, because we couldn't have the typical gamma ASO work here. Because we didn't want to reduce transcript expression, we actually wanted to modulate transcript expression. This required a fully modified backbone. This is because what we actually do is we sterically block, so actually it's a physical block that the ASO causes so that other proteins cannot bind to that region. And what this does is it changes the balance in how exons and introns are recognized. And in this case, basically trying to promote the inclusion of exon 8 opposed to its exclusion. So a lot of proof of concept was needed to see whether this could be even possible. So previous academic work, although done in a very small scale manner, showed that this was actually possible. So here we have 3 ASOs that were designed. They were transfected into the DYRK1A patient fibroblasts for a period of 24 hours. And what I want to bring your attention to is that we really do want to see an increase in the wild type bar, which is the blue bar, and a decrease in the mutant, while maintaining the total level of DYRK1A. And you can see that for a number of these sequences, that's exactly what we did. We had an increase in the wild type, or we had a decrease in the mutant, and no real effect on total DYRK1A. So this proof of concept was enough to basically characterize the mutation and potentially slightly de-risk the therapeutic strategy that ASO inclusion could work therapeutically. So this is now where n-Lorem's high throughput discovery pipeline comes in for this truly N of 1 mutation. Obviously, this mutation would never be commercially viable, so n-Lorem has the tools to do this in a rapid, safe, and ethical manner. So I'm just going to walk you through the pipeline. It starts with whole genome sequencing and ASO design. We designed around 370 distinct ASO sequences for this case, and this was done in-house through AI-informed design. And then it progresses through to our fully automated high-throughput ASO screening. And this starts at single dose, which is where we take all the ASOs and dose our cells at one single dose of 20 micromolar to identify any ASOs that are responsive in the way that we would like. So obviously, as I said before, increasing the wild type while decreasing the mutant. From that, candidates that show modulation go through what's known as a dose response, and this is a 4.5-fold serial dilution upwards to 20 micromole. And this shows us the range of which our ASOs are potent, efficacious, and selective. Then all of these ASOs are taken into what's known as a BJAB assay. This is done at a single dose to test for the activation of the innate immune system. And any ASOs that are targeted as inflammatory are then disregarded from further development. From that, we should have some candidate selections. And as I said, we should have some potent, efficacious, and safe ASOs that are ready for in vivo studies. So now I'm going to take you through the actual data of this pipeline. So I'm going to go through those steps: single dose, dose response, and BJAB assay. So I'm not going to show you all the ASOs that we screened for brevity, but what I do want to show to you is a region that was really responsive. And as you can see, they're all responsive to varying degrees. But what I'd like to highlight is this box over here. As you can see, what's happening here is we're getting a nice increase in the wild type expression while we're getting a good decrease in the mutant expression. So these candidates were progressed. Now you may be wondering why there's a region here that's doing the complete opposite. Well, this was actually designed to do this in mind, because when it comes to splice switching or splice modulation with ASOs, theoretically, if you can include an exon, you should be able to exclude it. So these ASOs were designed to actually skip exon 8 from the healthy allele as a form of control, and it also shows us the mechanism of haploinsufficiency. So here's our dose response, and as I said, it's in a 5.4-fold manner. And the bars, as they get darker, the concentration is increasing. And what we do want to see is a nice dose response. So the higher the dose, the more the increase of the wild type, and the less, more reduction, should I say, of the mutant. Obviously, splicing is a bit more nuanced than RNase H reduction. So sometimes it's not a perfect experiment whereby you get this nice dose response curve. But for some of them, they do look really nice. So for 31332, you can see here that we have a nice dose response for the wild type, as well as a nice dose response for the mutant, as well as a nice dose response here for 21331. So these ASOs are doing exactly what we want. When we increase the dose, we get a better effect. Moving on to our BJAB, again, I'm only showing a subset that progressed to further selection. But as you can see here, all of our test ASOs fell in the safe range. So we have 4 controls. One is a safe ASO that's known to cause no inflammation. We have then 2 intermediates that fall in this cautious range. And then we have an inflammatory ASO that if any ASO appeared above this threshold, it would have been immediately disregarded from further development. Now, in splicing cases, it's best practice to confirm the splice modulation at the transcript level has had a concomitant modulation of the protein expression. RNase H reduction is well known, and its downstream effect on the protein is very well characterized, but in splicing cases you do really need to check the protein, because as I said previously, it's a bit more nuanced. You don't always get a 1-to-1 ratio of transcript to protein upregulation. So in the case of our DYRK1A patient, we'd hopefully expect to see an increase in protein, and specifically an increase in protein in nuclear DYRK1A. So this is a Western blot, and I'll take you through this. We have here an untreated control. Here we have a non-templating control. So this is an ASO that doesn't bind, but it's used to take account for the electroporation that we do. And really what we want to see is the intensity of the bands be higher in our treated samples than they are in our controls. Now, what we did here, as well as an extra layer of analysis, was we fractionated the cells. And this is by separating the cytoplasm from the nucleus. You can actually separate the cytoplasmic proteins from the nuclear proteins and look at them independently. So the N stands for the nuclear fraction, and the C stands for the cytoplasmic fraction. And you can see here in the middle of the gel that we have a few ASOs that are really responsive to treatment, where you can see clearly compared to the UTC control that we've increased the intensity of these bands, both at the nuclear level as well as the cytoplasmic. Now, we did do what we call densitometric analysis, and this is using software to quantify the intensity of the band and then normalize it to those two other genes that, those two other proteins that you saw to determine how much we're upregulating it. So you can see that most of the upregulation is in the nuclear fraction, while we do get a modest increase in cytoplasmic fraction. We tested a few more ASOs in the same exact manner. Now we just loaded the cytoplasmic fraction first and then the nuclear fraction. But again, we have our two controls on the right-hand side here. And what we're looking for is that increase in band intensity. And again, in the middle of the gel here and towards the end, you can see that we've got some nice increase in protein expression. And when normalized again, you can see that there's a modest increase at ASO 21326 and some really nice increase in the nuclear fraction of 21330, 21333, and 21334. So in conclusion, academic and n-Lorem activities can happen in parallel for efficiency. And it highlights the need to really understand the consequence of a genetic mutation before we're able to take a case on. The n-Lorem pipeline leverages robots to ensure comprehensive design. And these robots also allow us to conduct high throughput, rigorous screening of ASOs in a time-efficient manner. All these processes are very scalable with high quality. So that's a good point to just stop on here. Although they're scalable and high throughput, we do not compromise on quality. And this is through decades of learning, decades of ASO experience, and obviously the help of automation. And lastly, it makes it efficient and more cost effective. And in the long term, that means that we can help patients quicker and faster without compromising any quality. Just before I say goodbye, I'd like to just acknowledge the DYRK1A Syndrome International Association for their grant funding that helped support a proportion of this work. And if you have any questions or you see me at the Colloquium, come say hi. Thank you for listening.